Hi, everyone. My name is Erin Hodgson, and I'm a professor and extension entomologist at Iowa State University. And we just like to welcome everybody for attending. This is our fourth year now of the regional webinar for soybean gall midge. And so, first of all, I'd just like to give some thanks to the University of Minnesota Extension. They're the ones behind the scenes helping us coordinate this webinar. So thank you, Phyllis, and thank you, Diane. You guys won't see them, but they're doing a lot in the background to make this webinar happen. And then you probably saw as you're scrolling through, we had many generous sponsors that makes our research and extension possible. So especially the University of Nebraska at Lincoln, the University of Minnesota, Iowa State University, and also South Dakota State University. We really appreciate all the support. And you probably saw a number of commodity groups, state groups, and then also the North Central Soybean Research Program has been exceptionally generous with this brand new pest we have in soybean. And then also a variety of industry sponsors that helps us put out really practical research-based type plots. And much of that is what we're going to be talking about today, some of the research going on. So, and then also we have a number of federal sponsors and partners, including the USDA, the North Central IPM Center, and SARE. So we want to thank those as well. You probably also saw as we're scrolling through the slides, our aggregated website where we push all of the events, distribution updates, other types of news related to soybean gall midge. So we want to make sure that you know about that, soybeangallmidge.org. And it's also a place where you can subscribe to the alert network. So if you're interested in getting some updates within the season, or if we have special events like field days, it can be a message that gets pushed to your phone as a text message, a voice message, or even an email. So it's a free thing that comes to you. We promise we won't do it too often, but just important updates. So you can sign up for that at soybeangallmidge.org. And if you happen to take a look at it recently, you'll notice that we've expanded in range for soybean gall midge as of last year, and we even added a new state, including Kansas. So now there's 7 states that have soybean gall midge and a total of 164 counties. And so we're not going to spend too much time on distribution today, but just to let you know that this pest has been expanding in range. What we're planning to do today is have really short updates on some research that's been going on within some of these cooperating groups. And we're going to sprinkle in some time for questions. And so we are going to just dive in to our first session here. It's about biology and ecology of soybean gall midge. And we have Dr. Justin McMechan from the University of Nebraska at Lincoln. He's an assistant professor and extension entomologist. And he's going to cover some of the adult overwintering. It's really important to understand for the summertime. And then also talking about some hosts. So thanks, Justin, for joining us. And I'll pass the mic.
Great. Thanks, Erin. Thanks for that introduction. So I'm speaking for a number of people on these updates because all of the states are involved that have significant soybean gall midge pressure in some part of this biology or ecology update. But this is a really brief overview. So if you find yourself a little bit missing in information, there's plenty of recordings on that soybeangallmidge.org website to look at. So really brief background, just if this is the first time if you're joining this, just to learn a little bit about soybean gall midge. It's a new species that's going to frame a lot of the talks today in terms of we're getting a lot of information, but there's a long ways to go. Those larvae at the base of the plant, they cause dark discolorations as they feed, and they essentially cut off the flow of nutrients. And the result is rather important under high infestation. We can see 18 to 31% yield loss on a whole field average if there's significant pressure up there and then 100% losses along field borders as indicated in the photo here taken in Nebraska. Erin covered this really well. We've got them in Kansas now, and her lab did a great job of picking up 6 new counties. Not that we want to find gall midge in new places, but we do want to know where it's at. And so none of this map represents severity. It just represents the presence of this insect. So, if we find one larvae in one county, that highlights the county. And so I wanted to lean in a little bit to a Nebraska soybean board funded project that allows us to check around 270 fields a year, just to show you what happened in Nebraska this past year. We got about 42 counties with larval presence, and that shading, the darker it is, represents the more larvae. If you look at the legend, and that lightest counties are the ones where we were in the county, but we didn't find any. And then if you look between that and wilting and dead plants, this is the bottom portion. We found about wilting plants in about 24 counties this year, really stacked up on the east. If we had more time, I'd show you previous years. But this year has been similar to the last couple years in terms of pressure and presence of the insect. Adult monitoring is a core component and a huge effort amongst all the universities that are participating in this. It takes a lot of effort from us, but it tells us a lot about the insect in terms of the challenges we're facing and the interpretation of some of our studies. Most importantly, and a take-home message from this is that they're all in last year's soybeans as we approach this spring. This is where they're sitting in those fields, maybe a little bit along the field border, but most are right along that in that last year's soybean field. So as you start thinking about this thing in a landscape level, that's where we seem to pick them up. We don't pick them up in fields that were soybeans 2 years prior. This is also a critical point is when we see first adult emergence. It's difficult to feel confident about what's happening with this insect in terms of the shift, but you'll notice 19 and 20 are kind of closer to mid-June, and then we've shifted back to late May and early June in these last 3 years. And why that's happening, not entirely clear, but whether or not we're getting better at tracking this insect for early emergence or something is actually shifting in the population remains unclear. This is probably the most important slide as we move through the different things in management today. I'd really like you to imprint this to memory. The duration of adult emergence from an overwintering site, the longer that duration is that they emerge, the more challenging it is to keep them out of our current year's soybean field. We're floating somewhere around 24 days. We kind of got into an ugly trend in 2021, and now it seems at least consistent for the last 2 years and how long they emerge from previous year's field. We'll be talking lots about different parts of the life cycle today. We have 3 larval instars that feed on the plant after it's infested. Then they drop into the soil, form these cocoons, and emerge as adults. And the adults are a big part of the focus in the spring for infestation. This is the 2023 season, and if you updated yourself on previous webinars, you'll notice that we saw them a little bit earlier this year, and the shift to peak was a little bit earlier. Typically, it's mid-June, but we started to see them in early June, peaking in population from overwintering, and then we got plenty of overlap at low levels. And then in mid-July, they started to pick up and actually stop before September, which is the first time that's happened. So last 4 minutes or so here, I'm going to really focus in on the hosts of soybean gall midge. Soybean, we know it's the focus of all the talks today for the most part. Alfalfa, I mention it because you'll see it as a host listed in various places. It's a pretty poor host. I wouldn't spend a lot of time on it. The only positive sample for that has come out of Minnesota. We thought we had some in Nebraska. They ended up not being positive. So be careful of the larvae that you see. Not all may be soybean gall midge. So feel free to send them our way or with the people in your state so you can see if they are soybean gall midge or not. Sweet clover, at least for Nebraska and Iowa, has been a very productive host for soybean gall midge. Not that it needs to be controlled, but we want you to know what we're seeing on it. Bruce Potter and that group, 2 different species of Phaseolus, and Bruce is on to answer all those questions. And then lead plant most recently. And if you're wondering, what is possibly unique about lead plant and its recent discovery is it's the first native host that we have found this insect on in this whole list. The rest are either Central, South America, or overseas elsewhere. For sweet clover, we have 2 different types, yellow and white. Whether or not they're a unique species is somewhat debatable amongst plant taxonomists. We can treat them as one or separate. Regardless, they have different durations of when they are productive host for soybean gall midge. Yellow sweet clover tends to peter out a little bit earlier. White sweet clover hangs around a lot longer. But regardless of that, we're picking up about 50 plus per stem. They vary across that geographic range that we're seeing them in. So up in Minnesota, we're not seeing as much, and Bruce has been really, surveying a lot and not seeing them. So we know we're not seeing as many up in that area. The few times when we caught adult emergence from them, it's pretty similar to what's occurred on soybean. We can move them from sweet clover to soybean and infest, albeit at low levels. And they have sometimes a dark discoloration or they're just gray in color, and the larvae can be well up the stems versus soybean, which is near the soil surface. Lead plant's an interesting host. We've looked at it a lot over the last couple of years, and this is the first time we've been able to find larvae on it. It is also unlike soybeans that the larvae are found right near the last part of the woody portion of the plant as you transition to green. And so this is a plant worth looking at if you're in an area where you don't have soybean gall midge. It takes a lot of work to potentially find them. And they were found within small wounds or cracks on those plants. We did the genetic comparison. They do match soybean gall midge, so we're pretty confident we're looking at soybean gall midge. And then Phaseolus, again, this is Bruce Potter's work up in Minnesota. They had a really nice sentinel plant system. 2 different species were used in that, lima bean and common bean. Infestation rates varied amongst the 2 hosts, and they had soybean as a check against this as well. And so a little lower on the common bean, but you can see that those number of larvae are quite low relative to, say, the conversation on sweet clover. And so I don't think we're seeing any plant death or real concerns, but these are worth keeping track of and monitoring, and certainly feel free to ask questions to Bruce. How does this all stack up? Rosiliella is an interesting genus to look at. And usually what we see is they're limited to plant families. So here we have a tree of the family Fabaceae that includes soybean and the hosts we've been talking about. For the other species that are in the genus Rosiliella, about 39 of them just have a single genus they're on. So quite a bit less, or 2 plant genera. So the limited host range we're seeing with these other species could be due to a lack of investigation, or maybe it's a reality, in which case soybean gall midge at 5 genera is quite large. So to summarize, the larval presence of plant injury is still abundant in counties with historical presence of soybean gall midge. And we certainly, I know Erin said they saw quite a bit in Iowa this year in the western part of the state. We've observed a shift to earlier emergence, but don't know why that's occurring. And the host range encompasses about 5 genera of plants within the family Fabaceae. Large populations are known to occur on soybean and sweet clover, but not so much for the other hosts. Thank you for your time, and take any questions in the transition.
Thanks, Justin. We had a good question about sweet clover. It is a common plant to see in the ditches in Iowa, maybe other states as well. Do you think that is contributing to low-level hosts or persistence of midges near those agricultural areas?
But potentially, I mean, that's a great question. I think people see a lot of yellow and white in the ditches, and for us in Nebraska, there's quite a bit as well. I don't see it being a contributing factor to risk of soybean fields. Erin, I see you nodding, so I don't think either one of us feel that way. It may provide background population, but with the abundance of soybean in our areas, soybean is contributing far more than sweet clover. So it's a little bit irrelevant when we consider just the amount that's moving around from our soybeans.
Yeah, I would totally agree with you, Justin. Thanks for your question. I did forget to say, and you announced it, Justin, that Bruce Potter is in the background. He's an IPM specialist at the University of Minnesota. So if you have questions, maybe for Southwestern Minnesota or anything related to maybe some of the alternative hosts, know that he's in the background. And then also kind of in the background is Bob Cook. He's a professor and extension entomologist at the University of Minnesota. And he'll be available as well if you have specific questions for Bob. But we are, I think, going to move on to our second presentation here. It is Pheylan Anderson. He is a master's student advised by Bob Cook at the University of Minnesota. He's a multiple award-winning presenter. We just came back from our regional entomology meeting, and he took home a really nice prize. So he is well suited for this kind of update, and a really interesting topic, talking about cold tolerance of soybean gall midge. So Pheylan, I see you there. You are ready to go.
All right. Hi, everyone, and thanks for that nice introduction, Erin. So today I'm just going to share some updates on my more recent research into the cold tolerance of soybean gall midge. There is a recording from last year with some of our initial work, but we do have some interesting new stuff to share this year that changes the story a bit. So let's hop right into it. So to start off, why are we actually looking at cold tolerance? Just a quick primer. Most insects are cold-blooded, so they have to rely on the temperature of the environment around them, which is a pretty major determinant of their range, especially in this part of the country where we usually have harsh winters. And in particular, actually understanding how a given insect overwinters and what the limits are of its cold tolerance, how cold it can get, how long it can be cold for, can be really important for understanding what areas may be at risk for expansion and what the populations might look like following winters. We know that soybean gall midge overwinters in the soil in silken cocoons, but we don't know what those limits are. So that's what we're going to be focusing on today. So for this experiment, we identified infested fields in our region, and we went down to collect a bunch of infested soybean kind of at the end of our growing season. So that was August 29th in 2023. And we collected all these infested stems along the field edge, so 500 in this case. And we're doing this because we want to study the overwintering stage, but we can't really go out to the field and actually get the cocoons out of the soil in the winter. So we need to actually trick them into overwintering for us in the lab. And the first step of doing that is tricking them into cocooning. So we took all those stems, peeled them open, retrieved these mature orange larvae. These are ones that would be dropping out of the plant naturally on their own soon. And we just put them in vials of sand that we had prepared. So from the insect's perspective, it just dropped out of the plant into some soil. It's going to dig down and cocoon. The next step is actually tricking them into thinking it's winter. So we stored those vials of sand at a simulated winter conditions, relatively mild, 37 Fahrenheit, 10-hour day length. And we kept them in there and then took some out after 1 month and 2 months of storage. And we also did a baseline measurement at 0 months before they went into storage. But what we wanted to record here is lethal temperature. And lethal temperature is basically the coldest temperature that an insect can survive a brief exposure to. And to figure out what that is, we picked these 5 treatment temperatures and randomly assigned our cocoons to be exposed to one of them, used a machine to cool them down, and when they reached the temperature, they were then pulled out, brought back to room temperature, and then an hour later, we dissected the cocoons open to see if the larvae inside were still alive or not. Let's take a look at the data we got from this. So on the y-axis on the left here, we're going to have the proportion that were dead by a given exposure temperature along the bottom of the x-axis. This is our first treatment, that one baseline before they even went into storage. And then these are the lines we saw for 1 and then 2 months of storage at the colder temperature. These lines track each other pretty closely. We did run some stats, which I won't go into a ton of detail on, but what they're basically telling us is that these lines aren't significantly different from each other. So it doesn't look like there's evidence of them acclimating as they sit in storage to these temperatures. What we can see by looking at the data, though, is a pretty sharp increase in mortality between this -4 to -13 Fahrenheit range. And in another experiment, we looked at the freezing points of these insects, and this dashed line represents that average freezing point, falls right into that range. This all mirrors stuff that we saw last year, so good repeating information there. So this is all good from the lab perspective, but what would they actually be experiencing out in the field? Well, we know that they would be buried in the soil. The soil is a good insulator. They would be experiencing milder temperatures for longer periods of time. But for our lethal temperature measurement, we actually removed them from the sand and we only exposed them to the temperatures for a brief moment. So it's not necessarily mimicking what winter might be looking like. It's just kind of a worst case scenario. So we think we can do better at that, and we're going to do this measurement of lethal time this year, which is basically pretty similar to lethal temperature, except you're looking at how long they can actually survive given temperatures. So here in 2023, we did this with the same field collected population, and we set it up in a pretty similar way with these vials of sand. We held the sand with the cocoons in it at 37 for 1 month. And then we kept some at that temperature as a control and then placed the rest at a just slightly colder temperature of 26.6 Fahrenheit. And then we checked on their survival pretty quickly at 3 hours as a baseline measurement, and then 1, 3, and 5 days. Let's take a look at our results here. Similar graph on the left on the y-axis, we're going to have the proportion dead, but now it's by a given time in days on the x-axis. This is our control treatment. As expected, they're doing fine out to 5 days, but things look pretty different for our colder treatment. And we can see that there is actually pretty high mortality after just 3 to 5 days sitting at this 26.6 temperature. So in summary, what did we learn from these two experiments? Well, we kind of repeated the results that we saw from last year, which is this -4 to -13 temperature range is significant. They seem to both die a lot and freeze a lot in that temperature range. And those two things are quite possibly linked based on that overlap. But they seem to be much less tolerant to longer term exposure. With just 3 to 5 days at this much milder temperature of 26.6, a lot of them are dying. For future work, we'd like to look at some regional soil temperature data. We did a bit of this last year, but we'd like to look at it in the new context of this new data. How often is it 26.6 for 3 to 5 days out there in the soil? We actually aren't sure yet, so that'd be interesting to look at. But I'd also like to share some other exciting new work that isn't directly related to cold tolerance, but you'll likely be hearing about more soon here. And that is we've started evaluating biocontrol with entomopathogenic fungi. Now that just describes a fungus which can infect and kill insects. Up here on the left, there's a soybean gall midge larvae in one of my sand vials that has been attacked and killed. And then on the right, there's an adult that we saw this happen to. There's some promising initial results. We're looking at several different strains. We're seeing maybe up to even 35% mortality in some cases. But this is really, really early research so far, so we can't really offer any specific recommendations on using this or using particular strains, but we will be researching it more. So stay tuned, and there will be more coming out eventually here. And I'd like to thank everyone involved and everyone attending.
Thanks, Pheylan. I know I get a lot of questions about overwintering survivalship in the winter. And I'm just wondering, how do you feel about the odds of surviving when the temperatures fluctuate? So it gets very cold, but then warms up. It's something that we saw a lot this winter. What does that mean for insects?
I think it definitely could affect things. There is some buffering that's going to happen because of them being in the soil, but there can be kind of complicated interactions there where snow cover on top of the soil affects how cold it gets. So I would guess it definitely has an effect, but it's not something that we've been able to actually test yet.
Okay. Thanks, Pheylan. We've had a couple questions about placement of the eggs within the soil profile. Justin, do you want to comment on some of the projects you've done? Just rough estimate?
Yeah, never detected any eggs in the soil.
I'm sorry, cocoons. Sorry.
No. I think it was actually an egg question that came through, so I think you repeated that question correctly. But yeah, we don't detect any eggs in the soil, just the larvae in various stages, depending on like third instar or in cocoons. Most are in the top 1.5 inch, but we have detected them all the way down to 6 inches or 5 to 6 inches. But we don't know if they can emerge from those depths.
Okay.
And that would be Mecca de Silva Lima's work with a USDA grant that we did that.
Okay. Thanks, Justin. Okay. We're going to switch topics now and move into biological control. Pheylan just hinted at it briefly with maybe some of the fungi that attack midges. But we're going to hear first about some predation that's going on with soybean gall midge. She's also an award-winning presenter, especially at our last bug entomology meeting in Fort Collins. She is advised by Bob Cook. You're sensing a theme here. He has very successful students coming out of his lab. So Sarah Lisak, she's a PhD student, and I can personally attest to blood, sweat, and tears on some of these projects. So, Sarah, could you share maybe some of your research with us today?
Yes, thank you, Erin. Hello, everyone. I'm going to be talking to you about biological control. So just to jump right in, we have, oh, it's frozen for a second. There we go. Okay, so what is biological control? So it's the use of living organisms or natural enemies to suppress pest populations. So these natural enemies could be predators, parasitoids, and pathogens. And today I'm going to focus on predators and parasitoids, starting off with predators. So these are organisms that kill another organism for their food. And we're going to focus on a ground-dwelling predator that looks promising as a biological control agent for SGM. We chose to look at the ground-dwelling predator as SGM is most exposed to predators in their larval stage, so when it falls to the ground, and its pupal stage, when it spins a cocoon and pupates in the soil. So the beetle that we're focusing on is called Pterosticus melanarius. It's a common ground beetle and a key predator in soybean fields. And previous studies have found this beetle to be the most prevalent predator in Minnesota soybean fields. So here is a map showing the distribution of this beetle. You can see it's all throughout the northern part of the United States and up into Canada. And if we compare that to our map of reported SGM, we see that it overlaps pretty well. So this distribution shows us that this beetle can prey on SGM within any of the states that you can find SGM in. So next, we're going to look at some lab assays to understand a little bit more about how the beetle behaves around SGM in the lab. So we're going to look at three main questions. So the first question is, do beetles eat SGM larvae in the lab? So beetles were collected from the fields, put into petri dishes with soybean gall midge larvae, and then which those were dissected out of the stems. And then we observed them to see if they would feed on the SGM larvae, and we saw that yes, they do feed on the juicy SGM larvae. And then if we get into our next question, do these beetles eat SGM pupae when they're protected by a cocoon? So this was to determine whether the beetles could recognize that the cocoons had food inside of it. So again, we had petri dishes where we set up beetles and we put cocoons of SGM in there. And we saw that, yes, they also will eat the pupae. So when observing the beetles, it took a while for them to actually figure out that there was food in the protected cocoon. But once they did, the beetles would quickly open up the other cocoons and feed on the pupae. And then our last lab essay that I'm going to go over, we have the question, do beetles prefer SGM larvae over other potential prey? So for this experiment, we put beetles in petri dishes again, this time with SGM larvae, and then also soybean aphid to see if they would prefer SGM over the aphid. And we saw that they do actually prefer soybean gall midge larvae over aphids. So potentially, if they were to come into contact with both of these prey in the field, it's most likely that they would probably feed on SGM larvae. Okay, so to summarize those results, we know that this beetle feeds on SGM larvae in the lab. They also can feed on SGM cocoons in the lab, and they prefer SGM over soybean aphids in the lab. So that's all great, but that's all just in the lab. So we have our big question of today of, do beetles eat SGM in the field? Are they actually going to seek out SGM and feed on them in the field? So this is where the gut content work comes into play that I've been working with, and it's using molecular tools to detect SGM within field collected beetle guts to detect for the presence and absence of SGM DNA in the guts. So to do this, we collected beetles via some pitfall traps in 2021 and 2022. And you'll see that there's this pink antifreeze at the bottom of these pitfall traps, and this was used to kill and preserve the beetles. And then the beetles were taken and frozen. And then we dissected out the beetle guts. And then all of those guts we screened for SGM DNA. So this was using molecular tools, again, to detect the presence or the absence of soybean gall midge DNA in the guts. So if we detect it, that would mean that they have fed on SGM in the field. So here we have the 2 years that we were working with, where we collected the beetles. On our bottom axis, we have our dates. And then here, our dashed line is going to represent our percent beetles positive for SGM. And so just seeing these dashed lines go up and down shows us that the beetles are eating SGM, and it varies throughout the season. So we don't just have a straight line across. It's not consistent throughout the season. And so we had the question of, well, why would this vary? And so we decided to compare it to the average number of SGM per plant, and that is going to be represented by this solid line here. So looking at the solid line, we see that it follows the general pattern of the dashed line. And this shows that there's a high correlation between the average number of SGM per plant and the percent beetles positive for SGM. So this tells us that the beetles are feeding on soybean gall midge in the field, and that they'll feed on more soybean gall midge when more are present. So the significance of these results, this beetle may be promising as a biological control agent for SGM. And it would be beneficial for growers to try to conserve natural enemies like this beetle through conservation biological control. And this adopts practices that helps conserve the natural enemy populations that are already present where SGM is found. And 2 ways to encourage beetles: you can apply insecticides only when needed. So this will minimize the killing and harming of beneficial insects like the beetle. And you could also focus on reduced tillage. So this will create hiding spots from the debris that's left over for the beetles, and also minimizes ecosystem disturbance within the fields. And then lastly, our knowledge gaps. So we only assessed 1 field in Minnesota, and that's just one part of a giant distribution of SGM. So future work needs to explore the predator community in multiple fields and in multiple states. All right, and that's what I have on predators. I can take any questions before moving on to the next section of parasitoids.
Yeah, thanks, Sarah. We had a good question from the gut content work that you're doing. Are you able to tell how much of the gut has soybean gall midge versus maybe other prey?
Yeah, so we didn't actually look at that. We were just looking at the presence and absence of the DNA. We could. It would take a little bit more work to quantify how much DNA is actually in there from soybean gall midge versus all the other gunk that's in there. We haven't looked at that yet, though.
Okay, but it might be possible. Maybe another person's dissertation.
Right.
Okay. All right. Sarah also has the next few minutes, and after that, we're going to have just a 10-minute break for people to ask questions that didn't get answered, or maybe other questions we're not directly talking about today. But in the same avenue, she's continuing the biological control, and this time shifting gears and talking about parasitoids. So take it away, Sarah.
All right. Thank you, Erin. So first off, what are parasitoids? They are insects that will lay eggs inside or on top of a host, and the eggs will hatch and feed on the host until it kills the host and emerges as an adult. So our lab has previously identified a wasp known to parasitize SGM. This is a new species and is known as Sinopius maximum. Wasps like this one are known to live inside of the host, so the eggs are implanted inside of soybean gall midge, and they live dormant until the mature larva or the prepupal stage of the host. So they wait for the SGM larvae to get nice, big, and juicy, and then will start growing and feeding on the host. So there are two main objectives I would like to address today. The first, to characterize parasitism spatially in 2021 through 2023, and the second, to identify the relationship between host density and parasitism. So we'll go into some materials and methods. So for the last 3 years, our lab has been exploring the parasitoid community. The sampling has occurred in 2021 through 2023, and in up to 17 fields and 4 states. So Minnesota, South Dakota, Iowa, and Nebraska have been looked at so far. And sampling occurred every 2 weeks once the field was identified as infested with soybean gall midge. So the number of samplings varied for each field. We collected 20 random stems on a field edge within a 50-meter distance, and then dissected out the SGM larvae and preserved them in alcohol for molecular methods. So we collected over 20,000 SGM larvae from the 4 states, and this was way too many to screen using molecular methods. So we developed a subsetting system where for low densities, where we had 0 to 8 SGM per stem, we sampled all of those larvae. And then anything that was higher than that, the higher densities, we sampled at least 10% that you can see here in this table. And that's just to be representative of the soybean gall midge in the stem. So then we screened the SGM larvae for wasp DNA. So this is using molecular tools to detect the presence or the absence of wasp DNA in SGM larvae. So basically to see which larvae were parasitized and how many. If we go into our results, starting off with objective 1, to characterize parasitism spatially, we'll go through each year. So in 2021, we assessed 1 field in southwest Minnesota. You'll see here on the map, we have orange circles for positive detection and gray circles for no detection. And you'll see that we did detect parasitism in this first year, and the season-long parasitism rate found was less than 2% of all SGM were parasitized. Moving on to 2022, we expanded the number of fields and the number of states we assessed, going up to 10 fields and in 3 states, so adding South Dakota and Iowa. And we detected parasitism in 3 of the 10 fields, but you'll see that was detected, parasitism was detected in all 3 states. And the highest season-long parasitism rate found was up to 4% of all SGM were parasitized, depending on the field. And then 2023, we added Nebraska to the mix. So we assessed 17 fields, and we detected parasitism in 8 of the 12 fields that have been assessed. So you'll notice the fields in Nebraska have not been assessed quite yet, but that is underway. So look out for updates about that. Our highest season-long parasitism rate found was now up to 7.5% of all SGM were parasitized, depending on the field. So to summarize all of those results, the highest season-long parasitism rate got up to 7.5%. However, each field varied in the number of SGM-infested stems and the density of SGM in a single stem. So this leads us into objective 2, which is to identify the relationship between host density and parasitism. And for sake of time, I'm only going to look at the data from 2023, this last year. And so to answer this objective, I have 2 main questions. The first question, does SGM density impact the presence of parasitism? So to look at this, on the x-axis, we're gonna have parasitism present or not, either yes or no. And then on our y-axis, we have the number of SGM larvae per stem. And so this is what the data looked like. And we can see that there's a significant difference between the yes and the no of parasitism present or not. And this tells us that stems with higher SGM density are more likely to have parasitism. And for the next question, we decided to focus on only the stems with parasitism present to compare the relationship between SGM density in a stem and the number of parasitized larvae, which is this question number 2. What is the relationship between SGM density and parasitism rate? So here we have the number of SGM per stem. On the x-axis and on the y-axis, we'll have the proportion of SGM parasitized. And this is what the data looks like. And we'll see that it shows an inverse relationship between the density of SGM and the proportion parasitized. And it was a significant relationship there. So this tells us that stems with higher density of SGM have a lower proportion SGM parasitized. But what does this all mean in terms of biological control? So this could indicate that parasitoids are limited by their egg supply or by the time it takes to parasitize a host. This could create stability between the populations of our hosts, so SGM, and the parasitoid, so the parasitoid doesn't kill itself off. So in summary, we have stems with higher SGM density. They are more likely to have parasitism present, but have a lower proportion SGM parasitized. And again, that just may be a limitation by egg supply or time. Further research is needed to learn more about that. And moving on to the significance of the results, these results help us to better understand the parasitism behavior of this wasp, which then informs the development of IPM programs with this wasp as a potential biological control agent. And some future work, we plan to repeat our screenings for parasitism in 2024, so this next year, and also analyze the relationship between parasitism and SGM density in other years to see if that trend is not just found in 2023, but also found for other years. And then lastly, some exciting news. We have found a new wasp. So from emergence cages in Nebraska, we had many wasps that looked just like this. This wasp comes from the genus Sinopius. We don't know what species it is, but it's the same genus of the one that I was talking about previously, Sinopius maximum. You'll see that this new wasp has a long, skinny abdomen, which is very different than our Sinopius maximum. So we plan to work with taxonomists to identify this wasp and also screen for parasitism of SGM from this other wasp, just like how we have been doing for Sinopius maximum. So we don't know if this wasp parasitizes SGM quite yet, but we are looking into that. And thank you all, and I will take any questions.
Thanks, Sarah. There was a question that came in about your predation study, the first part you talked about. Do you have any idea about the carabid larvae? Are they potentially having any type of impact on midges?
Yeah, great question. Potentially, yes. So we have not looked at the larval stage. We've just been focusing on the adults, but we should do some research on the larvae because they're in the ground, in the soil when the larvae are also in the soil. And so they probably come in contact with each other and probably are feeding on it, but we do not know.
Okay, so maybe a good chance of interacting with each other.
Yeah.
Yeah, we also had a question come through our Q&A box, and they said, or they were asking if there is any type of relationship or impact of wasp density with the proportion of midges that are parasitized. Could you speak a little bit more about that?
So how many wasps there are when there's the number of SGM higher in a field, the proportion parasitized?
I think so, yeah.
We haven't looked at that. That's a great question. We don't have records of like how many wasps are actually in the field at a certain time. We can only kind of take a guess of that based upon how many larvae are parasitized. that would be really interesting to look at. It would just be really hard to collect and see what wasps are even out there and how many there are. So
Yeah. Okay. Thank you, Sarah. We're just going to open it up for general questions. Some of you sent questions ahead of time, which is great. You can continue to add questions into the Q&A box. This might be a good question for Bob. Some of the Xenopus species that Sarah was talking about, is there any type of, could you say if they're the same or maybe related to the parasitoid wasps that we see for soybean aphid?
Yeah, so, these are all parasitic wasps, so they're kind of broadly related to one another, but if you look more closely, they're very distinct species and even genera, so there's really no chance of overlap, like the soybean aphid wasps attacking Gall midge or vice versa.
Mm-hmm. Okay.
But one thing to keep in mind is all of these beneficial wasps out there are pretty sensitive to a lot of the insecticides that we're using, so what we're spraying to knock down aphid populations are very likely to knock down some of these good insects. So just something to keep in mind when we're making those management decisions. And, I think a lot of us would argue that, use of the threshold is a good way to allow these beneficial insects to do their job. And then if they're not doing a good enough job, then we can come in with the insecticides, right?
Right. Yes. Good point. So different communities of parasitoid wasps that kind of have their niche as far as preferred prey. So yeah, good to think about. Bruce, if you're still on, we were wondering if you could speak a little bit about some of the seed treatment work you've been doing. It sounds like lotion, but it's not lubriderm, not lubriderm. Are you around,
No,
Bruce?
yeah, no, I haven't done any work on that at all. I just put that, yeah.
Okay. So you're just letting us know that it's out there.
I'm just letting you know it's not happening.
Okay. Do you have any guesses about the efficacy of this product on any life stage of soybean gall midge?
Well, it's chlorantraniliprole, so I mean there is some activity on flies. I don't know about gall midge in particular.
Okay. Great. Thank you, Bruce. We had a question, and maybe Sarah or Bob could address it. The beetle that you mentioned, that you were talking about, is not native to the United States. Do you have any evidence that we have some native predators or carabids that might like to feed on soybean gall midge?
Yeah, one of our previous students who started this line of research on biological control, Gloria Meloto, for her master's thesis, she did a bunch of survey work in a couple soybean fields and documented various species of these ground beetles and other predators. We focused on this particular non-native species because it was the most abundant, but there were several other species of ground beetles out there. I think most of those are natives, and in some kind of simple laboratory studies in petri dishes, they were all willing to eat soybean gall midge, which I don't think is a huge surprise, right? Because they're just these kind of soft-bodied, juicy food items, right? And these are generalist predators that are picking off a lot of things out there. But we haven't done the follow-up work, with the molecular methods to try to confirm if they're actually eating them in the field.
Right. Big and juicy. I have a totally different idea of soybean gall midge larvae now. Thanks, Bob. We've had just a variety of questions about cultural control, things that are basically in the hands of farmers to maybe disrupt the success of soybean gall midge. Justin, I'm wondering if you could just talk about maybe some just general cultural control strategies that we're perhaps not directly addressing today, some guidelines for us.
Yeah, after the break, I guess two students will hit on Natasha and Pragya will hit on some planting date and as well as some hilling related strategies. And you and I have tackled tillage, which has been pretty challenging to get some good results from. Just not for sake of or lack of effort, but just because the insect is difficult to track in those areas. So Yeah,
beyond
those two
the
all.
topics that we'll address in the second half? Any things that we can share?
Yeah, I guess on the tillage front, between you and I, we've seen some numerical response where we till last year's soybean field and potentially reduce number, but never anything that's significant or essentially wipes out the population. They're still emerging, and sometimes on that little bit of evidence, we see an extension of rather early emergence. So it might extend the adult emergence period. So that's a little bit challenging. Elliot Knoell will comment on the next one on this year's soybeans and tilling that, and some interesting early work there. But mowing borders, that's also something he'll talk about. So I think a lot of those potential tactics are there. Yeah, and I noticed, Erin, in the chat that they got a question on cyantraniliprole, and
Yes,
I assume they're talking about is that Fortenza?
I think so. Yes.
Yeah, and yeah, we ran that study, and that is a scenario where we are learning about soybean gall midge. We didn't know as much as what we would have liked to have known then, and we left some blank spaces around the ends of those plots when we did that study, and they wrapped that study, and it just so happened some control treatments that didn't have any insecticide on them got hit pretty hard, and it artificially elevated a treatment that was in the midst of that study. And so to nobody's fault, it kind of gave an appearance that something might have been going on, and I think some people may have been looking at that. We repeated that study the following year and didn't see those effects, and so it's the fallout of information coming out over time, and us learning about the insect. So it hasn't stood out relative to any other seed treatment, and there's lots of seed treatment data in the previous presentations that we've given.
Thanks, Justin. I don't know how common it is to overlap Dectes stem borer with soybean gall midge. I know that in Western Iowa, the overlap is basically the same counties. You could have both pests at the same time. Do you see that in Nebraska?
Yeah, you and I got nailed by quite a few reports on the interaction. We saw it at NREC, but that overlap, that Dectes stem borer, which is a debatable insect in terms of its problems if plants remain standing, where it chewed underneath the petioles towards the top parts of the plants. Soybean gall midge followed it in, and we could find large numbers of larvae in the petioles, and in some cases saw a bit of top dieback. So I think people are concerned going into 2024 of that interaction. It may increase larval number in plants, and we've got a study in Nebraska with fipronil product that no grower can use, but we can use to wipe out the Dectes and see if that relationship is still occurring. But it's interesting we've not seen it for a number of years and then are starting to see it now.
Yes, and I'm still learning about Dectes, but would you say that it's also an edge effect pest? So you have these two pests sort of aggregated at field edge contrast?
Yeah, it can go quite a ways into the field. Dectes can, but its occurrence is highest along the field borders, much like soybean gall midge. So yeah, they're occupying very similar spaces in the field.
Okay. Yeah, that's I'm still learning, but that's my impression as well. Okay. Justin, are there other questions that you think we should talk about just more generally? I'm trying to look through the pre-list that was submitted to us.
Guessing somebody asked about the distribution in the field. That's one thing we didn't cover today, but we have covered in the past.
Yeah.
We can find soybean gall midge out to 500 feet from overwintering generation adult movements, and that's on I would call a moderate pressure field where we see death for the first 10 feet into that field, pretty significant. And so they are well beyond our 100-foot kind of area that we focus on for management for people that are trying things.
Yeah.
And so people can expect them further in, and we saw plant death further in. But whether or not we should be managing that whole space, I think that's still debatable. But just expect they can move well beyond that initial 100 feet.
Yeah. Great. Everybody had time to stretch, maybe refresh the coffee. Welcome back. We have 5 more kind of short presentations before we just open it up again for the rest of the time with general questions. Next up, we have someone brand new to the group. We're so excited. We're bringing in more entomologists with more or some really interesting expertise. And so I'd like to welcome and introduce Dr. Adrian Pekarcik. Sorry about that. I'm learning your last name. He's a USDA ARS research entomologist at the Brookings, South Dakota station. And he's going to be sharing some information about entomopathogenic nematodes. And if you are dealing at all or interested in managing corn rootworm, you may have heard about this topic. And we're going to learn about how maybe these good nematodes can manage soybean gall midge. So thank you so much, Adrian, for joining us. Can't wait to hear about your research. Go ahead.
Yeah, thank you so much for the introduction. Today I'm going to talk to you about susceptibility of the soybean gall midge to entomopathogenic nematodes. And for the rest of this talk, I'm going to refer to entomopathogenic nematodes as EPNs. So these are any nematodes that are Heterorhabditis species or Steinernema species. And for those who are unfamiliar, entomopathogenic just is a fancy way of saying insect killing. So these are microscopic roundworms that are naturally occurring in all soils that have been sampled on Earth except Antarctica, and they require insects for food development and reproduction. And so in their life cycle, there's one stage in the life cycle that is free living in the soil and that is always found in the soil. And this is called the infectious juvenile stage, or IJ. These IJs live in a state of arrested development, so they don't feed, and their goal is to survive and to find an insect to infect. Once an IJ finds an insect, it will then try to infect it by entering the gut. Once the nematode is inside, the IJ will release a special bacteria it has that rapidly kills the insect within 24 to 72 hours. And then these bacteria will flourish inside and kind of digest the insect from the inside. Once it dies, the IJ will then resume feeding and development. So it'll feed, become an adult, and then the adults will mate and reproduce. And their offspring will start feeding, growing, developing, and become adults. And they'll do this for 2 to 3 generations before food runs out. When they're out of resources, the nematode will then these IJs will then take back that bacteria, put it back in their gut, and then they emerge out back into the soil in search of a new insect to infect. And one infected insect can yield hundreds of thousands of new EPNs. So it's a really promising management tool. Additionally, EPNs have been evaluated for other gall midge species, and they've shown a lot of success. So the Swede midge, which is a pest of broccoli and cauliflower, EPNs in laboratory killed 90 to 100% of larvae. EPNs also suppressed adult emergence from soil and broccoli fields. And another study found that these EPNs could actually move into the gall that was protecting the larvae within 72 hours of application. So the fact that soybean gall midge is spending a portion of its life cycle in the soil during overwintering and pupation, and these EPNs are naturally occurring there, they are very likely to encounter one another. And so the objectives of my study were to evaluate the susceptibility of soybean gall midge larvae to 4 EPN species in the laboratory, and then determine whether EPNs reduce adult soybean gall midge emergence from the soil. So focusing on this first objective, we collected soybean gall midge larvae by collecting whole entire infested plants from 2 soybean fields in Minnehaha County, South Dakota. We stored them at 15 degrees Celsius and used them within 72 hours. When we were ready for the larvae, we'd peel back the epidermis and then transfer the larvae with a paintbrush to our bioassays. For our EPNs, we used 4 different species that were obtained from a USDA lab in Georgia, including 2 Heterorhabditis species and 2 Steinernema species. We evaluated each of these at 3 different rates in addition to a control that had no nematodes. The rates were 500 IJs, which is approximately a field application rate. We also did 2,000 IJs and 5,000 IJs just based on other studies. To do the bioassay, we took mason jars, lined them with a filter paper to retain moisture, and then we applied our treatments in 1 milliliter of water using a micropipetter. We transferred 10 third instars soybean gall midge to each jar and then put a lid on them so they wouldn't escape. And then we placed the jars in a dark walk-in growth chamber set at room temperature for 30 days. Every 3 days, we checked the larvae, and any dead ones we would remove, dissect, and determine whether or not they had nematodes inside of them. And so what this process kind of looks like, here on the left, you could see a soybean gall midge larvae being surrounded by EPNs. Once they get infected, they kind of start losing their color and they sort of stiffen up, which is different because they are very gelatinous almost. And once the nematodes go through those 2 to 3 generations, the larvae is pretty much transparent, and you can see all the nematodes just squirming around, and then they emerge out, which you can see kind of by these squiggly lines surrounding this dead larvae. So from our infectivity bioassay, after 30 days, we found that larvae subjected to the highest inoculation rate were more likely to be infected than at the low and the mid rates. So at 5,000 IJs per mason jar, we had over 20% more infection, whereas in the 500, which is the field application rate, we had a little over 10%, so nearly twice the infection rate at the highest rate. Now, when we look at the treatments within each of these rates, so here on the x-axis, in the white bars is our 500 IJs, gray bars is 2,000 IJs, and 5,000 IJs are in black bars. And we have our control, which had only water, and our 4 different nematode treatments. We see that across all the rates, our HBVS, which was Heterorhabditis bacteriophora, that nematode caused significantly greater infection rates than the control at every single inoculation rate. At our highest rate, the 5,000 IJs, we see that all 4 of our nematode treatments yielded greater infection rates than the control. So this was the first confirmation of EPNs infecting and killing soybean gall midge larvae, and similar to other gall midge species, Heterorhabditis bacteriophora was the most effective at all 3 of our rates. But just from observation, these nematodes really kind of seem to struggle to enter the larvae. We obtained about 20% infection rates at the highest, so there is a little more work to be done to understand what limitations are holding these nematodes back. We also found that the highest rate, which was 10x the field application rate, was the most effective. And gall midge species are known to vary greatly in their susceptibility to these nematodes. In one species, the Brassica pod midge, it actually was largely unaffected by EPNs. So the fact that we're getting up to 20% mortality is very promising relative to the Brassica pod midge. So now quickly looking at our second objective to determine whether EPNs reduce adult soybean emergence from soil. We took specimen containers, placed autoclave soil, 10 healthy third instars, and then topped that with more soil to prevent the gall midge larvae from jumping out. And then we applied 1 milliliter of water with either no nematodes, 500 IJs, or 2,000 IJs. And we didn't test the 5,000 just to a limited number of gall midge larvae. And then we placed a lid with a sticky card upside down to monitor adult emergence. So the adults would fly up and get stuck. And we did this over an 80-day period. We'd check them every 3 to 7 days to count the number of adults. And what we found is that after 80 days, only 1 out of 10 adults emerged from all of our treatment rate combinations. And we think that this is kind of related to what Pheylan was talking about earlier, that we needed to stimulate this overwintering period. We collected these late in the season, right before they were about to pupate. So we've since placed them in a cold room for several months. And in the near future, we're going to bring them back up to a warmer temperature to see if we can increase emergence. So going forward, we're planning to evaluate some nematodes in the field this summer to see if they will suppress adult emergence and then reduce the number of plants that get infested by gall midge larvae in the adjacent fields. And so we're going to isolate and identify EPNs that are already occurring in these fields because they're more likely to be familiar with soybean gall midge. We'll mass produce them and then apply them back and then see what happens. With that, I'd like to thank everyone who has helped us with this project, and I'd be happy to take any questions.
Thanks, Adrian. I was wondering why maybe some of the species are less susceptible to an infestation or invasion by the nematodes.
That is a great question. And unfortunately, a lot of those papers, they don't really have any reasoning behind it. I can say that in a laboratory, like in a mason jar on a filter paper, that is a much different environment than the soil or in a soybean stem. So that alone could be having some bias in the results we're seeing. Additionally, it could be a factor of body size. So some of the really early instar gall midge larvae, they are so small, they're about the same size as some of these nematodes. So they might just be too small for the nematodes to actually get into.
Okay.
And the heterorhabditis nematodes in particular are smaller than the Steiner nemas. So that could be one reason we saw greater infection rates in those. Additionally, they have different foraging behaviors. So the heterorhabditis species, these nematodes actually will seek out hosts and move towards them, whereas the Steiner nema ones tend to kind of sit and wait around for the insect to come near them before they do anything.
Okay. There were a few more questions for you, but I think we'll hold off until the end. And thank you very much. We'll transition away from biocontrol into cultural control. So again, these are things that maybe a farmer could do to encourage or discourage midges. And so I'd like to introduce Pragya Gupta. She is an award-winning presenter. This is a reoccurring theme. She's advised by Justin McMechan at the University of Nebraska and Lincoln. And we just found out she's very quickly wrapping up her master's degree and heading to Texas. So Pragya, we can see your slides, but could you do the presenter view, please? Yeah. There you go. And she's going to be talking about susceptibility of soybean to soybean gall midge during the growing season. So we can hear you and we see your slides. Thanks, Pragya.
Okay. So thank you everyone for joining. And I'm a master's student at the University of Nebraska-Lincoln working with Dr. Justin McMechan, and I will be showing some results. So as Dr. Justin mentioned in the start of the webinar that soybean gall midge starts to infest the soybeans by laying eggs at the fissures and cracks that starts to develop at the V2 stage. And the hatched larvae feeds on the vascular system, disrupting the vascular flow, which leads to wilted and dead plants. And field observations have shown that these fissures and cracks are present throughout the soybean growth. And since this pest can have multiple generations, it has a season-long opportunity to cause infestation starting at any growth stage from the V2, which makes this pest unique, and the severity of infestation can vary depending on the growth stage it starts the infestation. Hilling is a practice of placing soil at the base of the plants, and knowing that fissures are present at the base, Dr. McMechan and his group conducted a study in 2021 to explore hilling as a strategy to protect fissures at the base and see if it can limit the soybean gall midge ability to lay eggs and cause infestation. What they found was pretty impressive. The study showed that hilling was effective in reducing the infestation. And the picture here clearly demonstrates the value of hilling. All the empty patches are where no hilling was done, and a lot of wilted and dead plants were found. But all the healthy soybean rows were where the hilling was done. So we found that hilling works. The objective of my study was to evaluate how different fissure exposure periods can impact the crop components during the season. And this was done to understand the critical stages when soybean yield components are impacted by the pest. We conducted the study at Eastern Nebraska Research Extension Center in 2023 with a randomized complete block design with 4 replications. So for this study, all the soybeans were held with the Henniker machine at the V2 stage when the fissure starts to appear. And after that, the fissures were exposed by removing soil manually at different growth stages, starting with the V2 stage. So all these 7 fissure exposure stages were our treatments. I will try to explain this better. So for the first treatment, the V2 stage was uncovered the same day it was held. Thus, for the first treatment, the fissures were exposed throughout the season, which means that the soybeans were susceptible to soybean gall midge infestation. For the next treatment, the soil was removed at the V5 stage, exposing the fissures. So in this way, the fissures were uncovered at different growth stages. And with this delayed fissure exposure, the soybean gall midge had a reduced opportunity to lay eggs as the soil was kept longer at the base, protecting the fissures. And our last treatment was the control that from which the soil was never removed. So it has the fissures covered with soil throughout the season. Coming to the data collection, we collected the data for larval abundance every time an unhilling was done. And this was done by collecting plants from the outer 2 rows of the plot and dissecting these plants back in the lab and counting the number of larvae. Next, we collected the data for plant injury that started in late June at 7 to 10 days interval for about 7 weeks. And this injury score was given on a visual observation based on the number of wilted and dead plants in the field. And later we converted these scores into AUSPC, that is area under the severity progress curve. And lastly, we collected the data for yield components that we collected from the middle 2 rows by randomly selecting the 5 plants. And these 5 plants were evaluated for the number of nodes, the number of pods, seed count, and seed weight. Now, jumping into the results, what we found in the results was pretty neat and something definitely that should be considered while managing this pest. The results for larval abundance, so on the on the x-axis, I have all the soybean stages when the soil was removed, uncovering the fissures. So the results for larval abundance showed that there was a significant difference in number of larvae. We found that the number of larvae were higher for the V2 stage, in which the fissures were uncovered early in the season, compared to the control. However, we see that the fissures that were uncovered at the R4 and the R5 actually has lower larval number compared to the control. This might be because with the delayed uncovering, the soil started to get hard and compact, and as the soil was removed at the later stages, a thin layer of soil might have remained, which might have protected the fissures. And for the control, since it was held just once in the season, because of the environmental conditions like rainfall, some of the fissures might have got exposed, resulting in little higher number of larvae compared to R4 and R5. When we see for the injury scores, we found that the V2 stage, again in which the fissures were covered earlier in the season, and V5 had a higher injury score compared to the later fissure exposures. And this declined as the fissures were protected for a longer time. Now, if we see for the yield components, we found no differences in the number of nodes. However, if we see for the number of pods, soybeans that were uncovered earlier in the season, that is the V2 stage, has the lowest number of pods compared to all the later uncoverings of the fissure. Now, if we see for the seed count, again, the lowest number of seeds were found for the soybeans that were uncovered at the V2 stage. And coming to the seed weight, the V2 stage has the lower seed compared to the control. However, we see that the R4 and the R5, they are not the V5 and the R1 are not different from the control. However, we see that there is a numerical increase in the seed weight as the fissures were protected for a long time. So now if we see the overall effect on the yield, since the soybean stages that were uncovered early in the season had lower yield components, it was reflected in the final yield. And we saw that the V2 and the V5 had a lower yield compared to the control. However, again, we can see that as the fissures were protected for a longer time with soil, the yield numerically increased over the season. Now, in the overall summary, we saw that larval number declined starting from the V2 until the R4. For the injury scores, we saw that significant differences were found, which was lowest for the treatments at the V2 and V5. And for the yield components, the treatments in which the fissures were exposed at the V2 stage impacted all the yield components except for the nodes. And that impact was reflected in the grain yield that was impacted more with the early fissure exposures. So overall significance of this study, the study tells that soybean gall midge can impact the yield components from any time starting from early June until late August based on the fissure exposure. The study tells the window in which the SGM can impact the soybean, and from the results we saw that the soybeans can be impacted from any growth stage starting from the V2, so the management might be needed as early as possible in the season. The study tells how important role the fissures play in soybean gall midge infestation and plant injury. And lastly, the hilling can be a potential strategy for growers that are under the significant pressure of soybean gall midge infestation. With that, I would like to thank everyone, and I can take any questions.
Thanks, Pragya. We did have a couple questions. I'm not sure, maybe best for you or Justin. I'll let you guys decide. Do the fissures, are they happen to all soybean varieties, or is there some slight variation of appearance and timing with some of the soybean genetics?
You want me to tackle that, Pragya?
Yeah.
I know Pragya and Erin as well, and her whole group have been involved in these evaluations, and to be honest, probably better just to save it for that last talk. I'll show some pictures in there. They'll cover it well. Great questions, though. All
right, we'll save it for our last session. So thank you, Pragya. We will move on. We have a few more questions, but we'll save them till the end here. Next up is Natasha Umezu. She is a PhD student at the University of Nebraska, and she has an award-winning presentation. You can tell these soybean gall midge topics took home a lot of hardware at our recent branch meeting. She's going to be talking about planting date and seed treatments as a management strategy. So Natasha, we can see your slides here if you turn on the presentation mode, and we can see your screen. So you are ready to go.
Okay, let's see.
Wait, hold on. It looks like it's in preview. There we go. You are set. Take it away.
Okay. Thank you, Erin, for the introduction. And hello, everyone. As Erin mentioned, I'm a... Oops. What's going on? Sorry. Let me see. It's something happening. Okay, good. Now we are good again. So as Erin mentioned, I'm a PhD student working with Dr. McMechan. And now I will be sharing with you what we have learned from our study about planting date and seed treatment. So before I move to our study, first, I would like to provide some information that will be important to understand the reason that we conducted this research. So here in this graph, we have the adult emergence from the overwintering generation. And as was mentioned before, the overwintering generation is the population that comes from the last year's soybean field. So in other words, it will be the first adult emerging in the season. And generally, the adults, they will start to emerge between late May to early June. And then there will be a peak of emergence between mid to late June. And then the number will start to decline as we move to July. So here, I just want to highlight the importance of the soybean growth stage in the soybean gall midge infestation, where at soybean at V1 would be considered as not susceptible since fissures are not present yet. And V2 is when soybean starts to be susceptible since the fissures start to be present in the soybean plant. And when soybean is planted in early season, plants at V2 can already be present before the peak of emergence occurs. So the time that soybean gall midge can infest would be longer when we compare to soybean planted in late season. So in this case, the duration of plant susceptibility would be shorter. So since we know that soybean growth stage is an important factor for soybean gall midge infestation, and in Nebraska, seed treatment has been used to control other early pests, we wanted to evaluate the effect of planting date and seed treatment on soybean gall midge larval abundance and plant injury. So this study was conducted in 2021 and 2023 in eastern Nebraska. And in both years, we had 5 planting dates, starting in late April until early June. And we had an interval of 10 days between each planting dates. And for seed treatment, in 2021, we used the rate used from farmers. But then since the seed treatment did not have a significant impact, we decided to increase for the highest rate in 2023. And because of the time, I will be very brief about how we collected our data, but you'll see it will be really quite similar from what Pragya presented. But if you have any questions regarding to this, please send the question to us and then I can answer later. So to track the adult overwintering emergence, we placed those wood cakes in the field in the beginning of the season. And then during 3 times of the season, a total of 6 plants per plot were collected to assess the larval abundance. So we collected the plants in the field and then we brought to the lab for larval count. And we also collected the injury score, which was explained by Pragya in the previous presentation. That is a visual evaluation where we gave a score from 0 to 4, depending on the percentage of injury plants. And then in the end of the season, we harvest the 2 middle rows of each plot to collect the yield. So with the results from 2023, we found that soybean planted in late April and early May had the highest level of infestation. The numbers drastically declined in those soybean planted between mid-May to early June. And when we look the graph for plant injury, actually we can see what we would expect, right? Where in those plants which we found more larvae, which was soybean planted early season, resulted in a higher level of injury plants. So those soybean planted between mid-May to early June actually did not have or just started to have those fissures in the base of the plant when the adults start to emerge. So this resulted in a lower soybean gall midge abundance and less plant injury. And even though that we had less soybean gall midge abundance in those soybean planted early June, in the end, the yield from those soybeans planted late season was the same as from soybean that has more larval abundance and planted early season. But we all know that yield is not dependent in just one factor, right? Since we have other factors that can impact on the yield, like environmental conditions and plant physiology. So those things is important to be considered when we talk about yield. And here in this graph, I would like to highlight the yield for soybean planted in mid-May, where in 2023, it was our highest yield. But when we compare with the results from 2021, soybean planted mid-May had the lowest yield. But one thing that is important to understand here from these results is that in 2021, the adult emergence was 10 days later. They emerged 10 days later than from those adults emerged in 2023. And here we can see that this delay on emergence could significantly impact in the larval abundance and yield for those soybean planted in mid-May. And in the second graph, you can also see in the yield graph, you can also see that the seed treatment statistical way did not have significant impact. But if you look those soybean that had more insect pressure, which was soybean planted late April to mid-May, the seed treatment could numerically increase the yield. So as a summary from this study, we could see that soybean planted in late April and early May were more susceptible for soybean gall midge infestation, so it resulted in a higher level of larval abundance and plant injury. But even though that we had this difference between soybean gall midge abundance within planting dates, in the end, the yield from those plants with higher insect abundance and lower insect abundance was the same. But one thing that is important to keep in mind is that if you plant your soybean in early season, even though you still have the same yield, it's important to understand that since you have more insect abundance in your field, you can increase your risk for the next season. And about seed treatment, we did not see any significant impact to control soybean gall midge. So with that, I would like to thank you your attention, and I will be glad to answer any questions.
Thanks, Natasha. I'm wondering if you also looked at population rates with maybe different planting dates or different seed treatments.
No, we didn't. I mean, planting dates, we had another study where we conducted in different locations, and the trend was the same, where in later planting dates, what we found less abundance. But for seed treatment, I know our lab had conducted some seed treatment research, but I don't think I will be the best person to answer about
Okay.
this.
Thank you. Just another follow-up question. Why did you pick gaucho maybe over some of the other active ingredients?
So gaucho is a imidacloprid. I forgot to put in this presentation. And this ingredient active is one that farmers used to use here in Nebraska to control especially bean leaf beetle.
Okay.
So since gall midge is a new species, so we would like to try to combine some management that farmers used to do already and see if this would be effective for gall midge too.
Okay.
Erin, if I can elaborate on that a little
Yeah.
bit, we move between, we've done stuff with thiamethoxam, and I'm seeing that Lubriderm or Lumiderm popping up again. It's possible that Nebraska has tested that last year, but we only had experimental compound list, so it might be in that compound list. And so I can't really comment too much on that. I know we did see quite a bit of differences amongst sites, and that really speaks to Natasha's data. Whereas we move sites or move years, things vary quite a bit, and it would be great to understand why that is. But we couldn't add all those treatments into Natasha's study. She'd still be in the field.
Okay. Thanks, Justin. Thanks, Natasha. We are going to transition once again away from planting dates to maybe some strategies that some of you have been thinking about with mowing and tillage. And so I'd like to introduce Elliot Knoell. I think this is also his first time presenting on our regional webinar. He works in the McMechan lab. He's a project coordinator and has all sorts of skills. But he is going to be talking about maybe some of the effects of this grass field edges and soybeans. So thanks, Elliot. I see you on screen and I see your slides. You're ready to go.
Okay, perfect. Thank you for that introduction, Erin. Yeah, so I'm going to be talking about some of the mowing and tillage studies that we've done here in Nebraska, also in collaboration with Minnesota as well, with Dr. Cook's lab. So just to jump right in, just as our general review, we know that soybean gall midge overwinters as cocoons in last year's soybean field, and then late May, early June, we have adult emergence out of this field, and they infest an adjacent soybean or adjacent soybean field. And in between, usually in between these two fields, you have something like a road, or oftentimes you can have a dense vegetation strip, such as a waterway or a tree line. And over the last few years, we, as a lab, we've observed greater soybean gall midge pressure next to this very dense vegetation. And we've actually used this tactic to in our surveys to better detect gall midge. So with this observed greater pressure, we decided to develop a mowing study, which was conducted in 2022 and 2023, where we basically took 100-foot strips of a grassy edge in between a cornfield and a soybean field, and we mowed that out. So with this study, we basically at adult emergence started mowing this strip, and throughout the season, we continued to mow every 10 to 14 days to keep a portion of that strip mowed down. And then another portion was unmowed throughout the whole season. So you kind of get that dense vegetation. And with this study, we also sampled, we sampled injury plant, proportion of plants infested, larval counts, as well as yield. So just to jump directly into the results, what we saw for 2022 was that with the proportion of infested plants, we saw less infestation in soybean that were adjacent to that mowed portion of that grass strip as compared to a soybean adjacent to that unmowed grass strip. So that was exciting to see. However, if we look at larval abundance, injury, or yield for that year, we really didn't see any differences there. At least statistically, we didn't see anything. So to jump right into 2023, again, we didn't see really any differences among these treatments, these mowing treatments, in terms of frequency of infested plants, or proportion of infested plants, as well as larval counts. However, if we look at plant injury, which is just an AUSPC value, or it's a way to basically for us to measure cumulative damage throughout the season, we did see a difference here. We saw less soybean injury less soybean injury in soybean that were adjacent to that mowed portion of that grass strip. However, it's important to keep in mind that this year, the 2023 year, we had very low gall midge pressure. So you could see we reached about 600 AUSPC value. Now that's pretty low compared to some of our other study sites. And in comparison, some of the higher pressure sites, it's not uncommon to reach 2,000 to 3,000 AUSPC units. So it was relatively low, but it was great to see that we were still able to tease out that difference. And then in terms of yield, we still yielded pretty well for both soybean treatments. We did get a significant increase, statistically at least, in that soybean adjacent to that mowed portion. But it was just a very slight increase. I don't think it was anything of great note here. And then looking at Minnesota's data, so they unfortunately they had some unfortunate environmental circumstances. In 2023, they had a very dry year, so they had very little vegetation for that grass strip, and they had less than 1% soybean gall midge injury. So very low pressure here, and they also did not collect yield in this, and that was mainly because of site distance in that year. But looking at the infestation frequency, they didn't see any differences here. So they didn't see any differences in soybean adjacent to mowed or unmowed portion of that grass strip. However, you can kind of see a trend here. Soybean that were adjacent to that mowed portion of that grass strip tended to have lower proportion of infested plants, but again, not statistically significant, so we can't really say for sure what's going on here. Maybe under a higher pressure, maybe something could be teased out here, but they had a very low pressure. And then looking at larval abundance, they really didn't see anything here. I can't really speak on any trends going on here either, but again, very low pressure here in terms of injury. So that may be, maybe those differences could be teased out under a higher pressure. So now I'm just going to jump into what Nebraska did with tillage. So with our mowing study, we decided to include a tillage portion of the study, where we basically had 3 different treatments. We had a no-till where we didn't touch the ground. We had a disking where we just disked the ground before planting. And I guess just to reiterate, this tillage was in this year's soybean field, not where the cocoons are overwintering. So in this year's soybean field before planting. And then our third treatment was included disking and hilling. And you saw with Pragya's previous presentation that hilling is a great strategy. And we decided to hill after the overwintering generation had stopped emerging. And this was to kind of try to capture that overwintering generational damage. So that's one reason why we did this study. And we got really interesting results out of this study. This was really exciting to see. So in terms of proportion of infested plants, we saw less infestation in both the disking as well as the disking and hilling combination. So these two, although not significantly different from each other, they were different from the no-till. We saw a very reduced infestation here. And then this also translated over into larval counts. So we saw less larvae in this disc portion of the disc portion, as well as the disking and hilling. And now those two were significantly different. But again, we were expecting to see less larvae here in that disking and hilling treatment because of that hilling. But to see that disking alone reduced the larval count, that was really, really interesting to see. And then in terms of plant injury, this also translated very well. We saw a less injury in both of those treatments. And again, disking and hilling, we expected less injury. We were expecting some injury because we hilled after that overwintering generation. So we were still capturing that injury, but we saw very similar results with just that disking treatment. So that was great to see. Now, how does this translate to yield? We didn't see any significant differences here in yield. Not sure why that is. That may be because, again, we had a very low pressure site. And so hopefully under a higher pressure, we may be able to tease out those yield differences. We may see a yield difference there, because again, we had a very low pressure site in 2023, and that's just because of the location, unfortunately. So in summary, what we saw was that the mowing of that grass edge inconsistently reduced gall midge infestation, larval number, and injury, as well as yield in soybean. And one importance this dense vegetation may have in gall midge ecology is that this dense vegetation may hold on to moisture longer in the day. We've seen that from Pheylan Anderson, who spoke earlier, so from some of his data, that moisture is very important for adult survival. So it may play a huge role, that dense vegetation may play a huge role in adult survival in the field. And we've seen that moisture is very important for other Rosselliella species as well. One important thing to consider is corn development. If you have an early planting of corn, that corn is going to be more developed earlier in the season, which may kind of overshadow that importance of dense vegetation, depending on how dense that vegetation really is, as opposed to a later planted corn date. You'll have shorter corn earlier in the year, which maybe that vegetation will become more important. And one final point was that in 2023, that disking, we saw the disking of the soybean field, this year's soybean field before planting, reduced soybean gall midge infestation, larval number, and injury, but we didn't see any effect on yield here. But again, this was very low pressure. Maybe it'd be different under a higher pressure system. So with that, thank you, and I'd be glad to take any questions if you have them.
Thanks, Elliot. I don't see any questions directly related to mowing and tillage, at least at this time, but if you'll stick around, we might have some come in. Our last session, we're bringing it back to Justin, and he's offered to do something, I think, kind of a little bit brave. He's speaking on behalf of Dr. George Graff. He's a plant breeder at the University of Nebraska-Lincoln, and he's going to be talking about some of the host plant resistance, germplasm work that this collaboration has been spending some time on, and hopefully you can address some of the fissure and genetics variety questions that we have before. So we can see your slides. We see you.
Hey, great. Thanks, Erin. Yeah, I am not George Graff, so I'm going to do my best to cover his stuff for him. He's, I believe, down in Puerto Rico right now working away on other projects. So I'll do my best to summarize what he's got. This is one of the very exciting areas of research and kind of a gold standard for what we're after in terms of management of soybean gall midge. So we certainly didn't want to leave it out. Erin Hodgson's, Adam Varenhorst and I have been conducting these trials with George for the last couple of years. We're really just going to focus on 2023, and we've gone from 760 or so accession lines down to now about 100, and are toying around with lower numbers where we're getting more detailed data. But in this current year, we had 90 resistant quotations, those that showed less injury in previous studies, and 10 that showed plenty of injury in previous studies. And we put those at 2 Nebraska locations and an Iowa location with 3 reps each. We documented larval presence or absence in those. And then Mitchell Helton from Iowa, his scoring system, he was a graduate student of Erin's. We used that scoring system and took data every 10 to 14 days through R6, so very similar to previous work. We also selected a few accession lines from the previous year for a more detailed study. There were 18 of those. 16 were considered, again, quotations resistant, and 2 were more susceptible plus the susceptible check. Put those in 4 rows like our larger studies, and then we actually took counts, larval counts. We got questions about fissure types. We have seen different fissure types. We've seen kind of a surface fissure versus a deep fissure. And you saw some that look like they go way into the stem, and others that are just very, superficial. As well as the presence of those fissures either above or below ground. This was a pretty deep planting this past year for depth, and that led some of those fissures to be pretty close to soil surface, if not below, which is interesting in itself. Now we're going to pile right into the data on this. So we've got some accession lines here on the x-axis and plant injury on the y-axis. Not extensive values, but this is a combination of a couple sites. And so we're looking at a susceptible check that's on that right side, and then these potentially resistant lines that were expected to be resistant are showing quite a bit less injury, which is pretty exciting. I'm going to layer over this larval count. And so it tells you a couple things when you start to see larval count per plant. Those ones where we're seeing a lot of issues, we have higher numbers of larvae that we're finding. And then on the opposite end, we're finding very few larvae, but even as close as they may look to the x-axis, we found larvae in all of them in some plots. So they are present in all those plots, but at much lower numbers. If we now kind of bounce our way through the maturity groups that have been studied over the last couple of years, we have, again, those same locations, 9 reps now total. And you can see plant injury again. This is maturity group 0, so not something we're typically planting. And then those dots, again, representing larval presence. And so you can see at the higher end here, a lot more of those plots are infested because this is presence absent. So if all plots were infested, it would be a 1. Any degree away from that, and it starts to drop down. And it's a combination of all the sites that are involved. But you can see as we go down, some of those tend to drop, and then the proportion of plots that are infested at those different sites. Get into maturity group 1, we got a lot more numbers here. Again, each maturity group is showing a similar relationship where some of those accession lines within that maturity group are showing quite a bit less injury relative to that susceptible check. And larval numbers or presence is kind of decreasing as we move in a general trend away from those. As we get to maturity group 2, this is now, touching Nebraska, at least moving south, and obviously these are up in Minnesota with some lower maturity groups too. And you can see a lot of larval presence through this, but again, that drop is still occurring for maturity group 3 or 2. And as we get to maturity group 3, the same graphs that we're looking at, we're again seeing opportunity in this maturity group as well. Some showing more susceptibility than that check, but again, a general decline as we get to some that are showing less, as well as less larval, a number of plots infested. So this is that's all good news, obviously, looking at those last couple graphs, but these are accession lines. So for anybody who's not familiar with this, we don't pick these up and plant these in large commercial fields. They're a long ways from that process, but for soybean breeders and various companies, George intends to provide them with that information as we finish this project or this portion of it, and they could look at their own breeding. In Nebraska, we have some elite soybean lines that we've been testing for a couple years, and a good part is they're skewed towards less injury as well. So there's opportunity at things that are closer to release as well, and so this all points to a lot of potential in the future. And again, if we just look at the difference in maturity groups 2 and 3 across those, we're seeing similar responses on those maturity groups. We've got injury in all, but quite a bit less than that susceptible check. So in summary, we've got this differential response, that we're seeing with genetic material in terms of, what is the infestation. We've got genotype differences. There's some small graphs on the bottom from previous talks that have been given on this. We've got some significant SNPs to look at. And this validation process that we did this year is rather promising that we're continuing to move in the right direction. Those accession lines with very low or no AUSPC scores indicates that we're getting there. And I'll point out, because you've seen fissures in other presentations today, look at how light those fissures are. And so Erin and I have both seen this in studies. They're very light. They still get infested, and we need to take a closer look at those categories relative to infestation. But it's just in this photo, you can really see the difference in injury across these plots from what we're seeing in the field. So there's significant potential for resistance in the elite germplasm that's closer to release. That's a good news for farmers because that can be screened. But there's still quite a bit of work to done in this category in terms of the next steps. And so what are we doing? What's coming next? You're probably getting tired of hearing about this hilling thing, but it's a wonderful research tool, although a lot of growers can't use it. So the plan is to put in yield tests with those elite lines and hill half of them and see how good they really are compared to keeping gall midge mostly, or if not completely out. And we've got a lot of work to do in terms of the uniform test entries for genotype information and evaluating for selected lines for reaction in infested fields. And then we plan on taking those most susceptible or resistant lines, looking at them under control conditions, hopefully here at some point. And of course, we've enjoyed the low numbers. We'll probably head to some higher ones as we start crossing populations for further genetic studies. So with that, just a thanks and a thanks to the funding sources. A lot of Pragya and Natasha's work is also Nebraska Soybean Board funded, but NCSRP has put a considerable amount, as you've noticed them through the talk today, and just want to thank all of our partners on this.
Thanks, Justin. Hopefully those folks that had questions about fissures, their questions were answered. So we have just a bit of open time now, and I'm going to try and get to the questions that we didn't get to today. And there's a couple questions for Adrian about the nematodes. And Adrian, I wonder if you could speak to, are there any cultural management considerations a farmer could use or implement to encourage the nematodes to maybe stick around or even build up in numbers?
Yeah, that's a great question. And it's one of the reasons that we are planning to apply these nematodes to entire fields is because in general, it's sort of a needle in a haystack. You have a couple nematodes here and then a couple nematodes all the way over there and nothing in between. And their populations largely cycle with pest populations. So the more insects or food there are for the EPNs, the more EPNs there will be. And so in periods where you don't have a lot of food or something, then they're gonna just die because they've run out of food for an extended period of time. In general, fields with higher organic matter tend to have better populations, and this could just be related to soil moisture. These nematodes require moisture to move, and the moisture could be as little as relative humidity in the pore space in the soil. But yeah, those are two limiting factors, and soil type will also influence the ability of certain nematode species to move or not move. So heavier soils will be a little more restrictive for like Heterorhabditis species, but pretty much all nematodes do really well in lighter, sandier soils.
Okay. Yeah. Interesting. Thank you, Adrian. Pheylan or Bob, do you know about how many eggs a soybean gall midge female can lay? Or maybe you have an idea of maybe based on some other midges in that same genus?
Erin, that's a good question. We don't know specifically for soybean gall midge. We've been playing around a little bit with methods for that, trying to dissect out the ovaries from females and counting how many eggs are there, or seeing what we can get for egg lay on plants in the lab.
Okay.
But if we look at other species of gall midges, an individual female can produce, 100 or more eggs.
Okay. They'd be laying those eggs over time, you think?
Yeah, but most gall midges aren't very long lived either. Yeah.
Okay, so over time for them could be like 3 to 5 days.
Yeah, probably.
Okay. Okay. Thanks, Bob. Elliot, I'm wondering if you could provide a little bit more information about your tillage. What type of disc did you use? And do you have any thoughts about using vertical tillage as maybe a management strategy?
Sure. Yeah. So as far as the type of disc, I don't remember off the top of my head. Justin would be able to answer that.
Oh, okay.
Yeah, it's a disc harrow. So an Amco disc harrow is what we used. I ran around with it, so Elliot saw it go by really quick.
Yeah. Yeah, and as far as vertical tillage, I'm not too familiar with vertical tillage, but it's my understanding that it causes a less impact, right, to the soil. So not really sure why we're seeing the differences we're seeing in the disking treatments. Honestly, I think, correct me if I'm wrong, Justin, but initially what we did was we included that disking treatment because we wanted to isolate any disking effect that we would have with the hilling. Because we wanted to isolate that our initial thing that we wanted to do was isolate that overwintering pressure. And we just happened to get that difference with the disking. So I don't think we're really sure what exactly is going on. It may be, movement of the soil is causing maybe a loss of moisture, or something along the lines of that. Maybe it was just the location of the study and something environmental happened. We don't really know. So
Okay.
vertical tillage could help. I can't really say if it would or not, though.
Yeah. You haven't looked at it for yourselves.
Yeah.
Okay. Thanks, Elliot.
Yep.
Justin, while we have you on the screen, or maybe Natasha could also hop in. Do you have just a general best guess of like optimal planting date for interfering with success of soybean gall midge, just given a wide range of maybe spring conditions that we might see every year?
I would, Natasha can say
Yeah.
as much. I know the two of us have had a lot of conversations with farmers. This is a touchy topic, right? Yeah, you know that as well as everybody else that's on the call. We have generally recommended plant your high risk fields last, has been the take home from that. And I think people should realize that mid-May planting is going to fluctuate, depending on adult emergence. And I think that question about the growing degree days is a critical one. It's not one that we feel super confident in based on other species, raspberry cane midge overseas where they try to do that. And our capture network, although really useful right now, is not terribly accurate. And so we could use a pheromone, which again, Kesey's working on. Natasha, I know you've got a lot of thoughts or other things about this, but your thoughts on that topic.
You know, I agree with what Justin mentioned, and it's complicated because, again, since the duration of the emergence is so big, it's difficult for us to say to farmers, okay, plant in this day and then everything will be fine. This would be our dream. But as Justin mentioned, if you can delay as much as you can, would be, right, a good maybe option. But just to try to reduce the insect population during for thinking about next season.
Okay. So plant your highest risk fields last to try and maybe just minimize the chances of getting that infestation. Yeah, I agree with you there. Yeah.
I had one question one time asking if we could like plant later just in the border of the field.
Yes.
This is something that we never tested, but it's complicated because, as Justin mentioned, we can have soybean gall midge going to the center of the field until 500 feet, so it would be complicated.
Yes, I get a lot of questions about planting a trap crop or just mixing up maybe some of the planting dates within a field to try and minimize it. But it always seems like there's new plants available at that time, if you're mixing up trap crops with other planting dates. So yes, thank you. Bruce, I was wondering if you're still on, do you have any ideas about the amounts of some of the beans, the phaseolus that are planted in Minnesota? Do you have any idea of like acreage?
Well, I don't, off the top of my head, I don't know the exact number of acres, Erin, but we have a pretty significant edible bean planting in particular in the western and northwest part of Minnesota.
Okay.
And part of that in west central Minnesota, where that little hump comes out to the west, the dry bean production and gall midge populations overlap pretty well.
Yeah, that was my thought, that Minnesota maybe had more of the edible beans or dry beans compared to other states that are infested with midges. So do you think it's impacting their production at all? Are they changing anything up?
Not at this point. We can find them there, but I think as Justin mentioned, the injury isn't as high as it is in soybeans right next door, that sort of thing. So at least at this point, it's there. It's something people need to be aware of, but that's about it.
Thanks, Bruce. Maybe Bob or Justin, if you have any final thoughts. We didn't really talk too intensively, or we didn't focus on chemical or insecticides. It's just a general management strategy. I'm wondering, what do you say to people that are wondering about like what to use or when to use, how often to apply? Do you have just general comments about chemical control?
Yeah, it's tough. And I run into people every fall after kind of the battle has already occurred. and I hear all ranges of responses to, I got the alert network, I sprayed. Usually it's some pyrethroid type product, so it contains a pyrethroid. Some of them say, hey, excellent control, I'm doing it again. There are some characteristics about what they're doing that we want to look at, certainly. Others, same occurrence, almost same timing, and really frustrated that things are not working. So I think anybody looking at managing the chemical side of this or using that as a tactic, you really need to be under a lot of pressure to be considering that. And then you need to understand that things may not go as planned based on other studies that you're seeing.
Mm-hmm. Thanks, Justin. Anything to add, Bob?
Yeah, just to comment that, this is a very different beast than soybean aphid, right? That was an invasive species that came in, but, fortunately, we've been able to manage that pretty well over the years with insecticides. Clearly, from everything you've seen, we don't have a silver bullet yet, and I don't know that we ever will for soybean gall midge, so we're really gonna have to look at true integrated pest management, right? So I think all these different areas of research, hopefully we can make advances on various aspects and start, cobbling them together into, true integrated plans, right?
Yeah, I totally agree. We still have a lot of interest. As new formulations or new actives are coming out, I think all of us are interested in screening that. But right now we don't really have a good, just generalized game plan to attack either the adults or larvae. So I just want to be mindful of time here. We've hit the end of our 2-hour webinar. I hope that most of your questions got answered and you learned a little bit more about the research we're doing here in this very large collaboration today. So we'll stick around for a little bit, but thank you all for taking time. You guys came from all over the place and we appreciate that. So thanks, everyone.
Erin, I guess as people are kicking themselves out and getting their CCAs, I noticed there's a question that I don't think I hit directly that was related to the fissure presence.
Yes.
And we find them on all accession lines we've looked at so far. I think your lab the same. So there's not too likelihood that we'll see soybean without fissures on it. You can never say never. Maybe it'll show up, but so far they all seem to have some type of fissure presence on them. And and manipulation via management of very high populations, even 600,000 plants per acre by mistake, still had fissure presence on them. So we're not likely to do that with those types of tactics either.
Yeah, when you really start looking, you notice, like you said, the variety, and like some are really superficial, some are just deeper inside to the stem. There's quite a variation of how a plant develops a fissure. Yeah, I guess there was another question about systemic insecticides, whether I suppose via seed treatment or a foliar application. Do you think how it moves through the plant will make a difference or could make a difference?
Yeah, this is more of that really fuzzy research that doesn't seem to be super consistent. We have done drop nozzles with neonic type products that are systemic, and we've seen shift in larval type between treatments, meaning that there was more white larvae over a longer period of time than orange. Pretty transient effect. It didn't last very long and it was gone, and it didn't contribute to yield differences at the end of the season. So I think it's that same type of thing. Thine that is one product that is systemic, that is granular at plant applications are made for that. And Erin, you and I have looked at that. It has quite a bit of efficacy, but it is not a tool that is easily picked up by farmers because of the specialized equipment on planters that are needed. And so it has its potential, but it has its challenges too.
Yeah. Bob, do you think there'll ever be BT soybean in the Midwest of the US and Canada?
That'd be pretty cool for something like this. I don't know that any of the companies would be jumping on it real soon, but if they do, I think it'd be exciting for us entomologists. There is a strain of BT that is effective against flies or fly larvae, right? There's a strain that works against beetles, a strain that works against or subspecies, whatever it is, that works against caterpillars and then a separate one for flies.
Are you aware of any BT that's incorporated into plant genetics for flies?
I'm not, no.
No, it's
Like I think they
mostly
use it for mosquito control and things like that, right?
like either sprays or like dunks or drenches. Yeah. Yeah, that would be a game changer. I have also heard absolutely zero interest in getting BT soybean in the US, but yeah.
There's a
Justin
comment
or Bob,
there.
oh, go ahead.
What about that? I'm not sure if this question that came in was addressed about kind of like getting your neighborhood to sync up your crop rotations to break the cycle.
Doing like an area-wide management. Yeah, what do you think?
I don't know if we know enough about how far this thing can really move, right? I mean, it seems lazy and just kind of hitting the edges of the next field, but
yeah,
if it really needs to, you know, who knows how far it can really move, or, you know, with the winds and things like that.
Yeah, the area-wide plan has worked for a few other really devastating pests, but more on I think it was a regulatory mandates for some pests. What do you think, Justin? Could we get people together?
Oh man, good question. In the survey where we see corn, soybean rotations, this thing slows down a lot. That's like so central Nebraska area. So, if places predominantly move to that, I'm sure it would probably pull its way down. Bob's right. It's hard. It's especially hard with the host range that we're seeing in sweet clover being so abundant that in some places where we've come to and later looked at CropScape and found there's no soybeans within a 1 mile radius around that entire sample, we can detect gall midge in the middle, but we're not certain if there isn't sweet clover or something else. We just know in those spaces, gall midge isn't a problem. It's present. It's not causing a problem. So that gets to Bob's point is, could they cause a problem if they're moving that far? And, is it just a fraction of the population that makes it there?
Yeah, it almost seemed like we'd have to introduce a little bit more crop diversity as well, to be able to do that. And there's pluses and minuses to doing that. But it would be a big stretch to have an area-wide program. It could probably benefit from other pest suppression too, right? Not just soybean gall midge, but by having more cropping diversity, it would probably lessen the overall pest pressure. That's a whole nother topic. I'm not sure how long, we have quite a few people still hanging on, which is shows interest of this topic, but I'm not sure how long we want to hang on and maybe just field some other questions.
Yeah, if people have them, I can certainly, I know some of us would stick around.
Yeah.
And I appreciate Kurt's comments. We've worked with Kurt in the past, and so he's familiar with gall midge in terms of some of the stuff. But yeah, it's just a challenge to that field rotation scenario between farmers. What I've seen in Nebraska, at least, is where a farmer maybe had a field split up, corn, soybeans, half and half. They merged a lot of those to just corn and then just soybean, and took out a lot of those smaller fields in areas where there was a lot of pressure. But to see it change on a landscape level, I've not seen, broader landscape level, I've not seen that.
Yeah, maybe Bruce has a follow-up to this as well. Go ahead, Bruce.
Well, I think it's if we're talking about the area-wide thing, I think we kind of already see that maybe with things like tillage and planting date. We just don't understand it or don't recognize it yet. I still think though that and I just don't have a spot to test this out. But it'd be really interesting to try this trap crop concept and even plant some soybeans into corn and then terminate those real early. See if that could knock down populations.
You got that covered. Again, thanks for joining. Thanks for sticking around into overtime. Appreciate it. And we will be in touch. So thank you, everyone.